Good morning, students. In the last class, we have discussed up to endoplasmic reticulum. Endoplasmic reticulum, it is of two types: rough endoplasmic reticulum and smooth endoplasmic reticulum. Right? The rough endoplasmic reticulum, it is mainly involved in protein synthesis, and smooth endoplasmic reticulum, it is mainly involved in lipid synthesis. Why rough endoplasmic reticulum is mainly involved in protein synthesis? Because ribosomes are present on the surface of the rough endoplasmic reticulum, whereas ribosomes are absent on the surface of a smooth endoplasmic reticulum. Right? What are ribosomes? Protein synthesizing factories. Protein synthesizing or protein manufacturing unit. Synthesizing or manufacturing units. So whatever it may be, both rough endoplasmic reticulum and smooth endoplasmic reticulum together are synthesizing proteins and lipids. Using proteins and lipids, biological membranes can be synthesized. This is called membrane biogenesis. What is membrane biogenesis? Synthesis of biological membranes using the proteins and lipids that are synthesized or manufactured on rough and smooth endoplasmic reticulum respectively. Now coming to Golgi apparatus. Golgi apparatus was first described by a scientist known as Camillo Golgi, who was born in Cortino near Brescia, like that. Some story is given there in a box-like item. Okay, right, and he has described about Golgi apparatus, dark reaction or black reaction. Okay, and he was awarded Nobel Prize also in the year 1906. All that entire story you can go through in that box. The Golgi apparatus is known as Golgi apparatus because it was discovered by Camillo Golgi. Camillo Golgi. In which year he was awarded Nobel Prize? He was awarded Nobel Prize in the year 1906. Nobel Prizes were started given from which year onwards? 1901. So these people are the early Nobel laureates. Okay. Golgi apparatus is also known as Golgi complex. Golgi complex. It is a system of membrane-bound vesicles arranged approximately parallel to each other in stack. Stack means like a bunch. Membrane-bound vesicles that are arranged. Membrane-bound vesicles which are arranged approximately parallel to each other, not exactly, approximately parallel to each other in stacks. System of membrane bound vesicles arranged in parallel to each other in stacks. Stacks. Membrane bound vesicles. The membranes of Golgi apparatus have connections with the membranes of endoplasmic reticulum. Membranes of Golgi apparatus have connections with membranes of endoplasmic reticulum, and by having connections with the membranes of endoplasmic reticulum, cellular membrane system is formed. Cellular membrane system is formed. How cellular membrane system is formed inside the cells? The membranes of Golgi apparatus have connections with the membranes of endoplasmic reticulum and form cytoplasmic or cellular membrane system. Cellular membrane system. In detail, you are going to learn about the cellular membrane system in your class 11th with a term known as endomembrane system. So, how the cellular membrane system is formed? Membranes of Golgi apparatus and membranes of endoplasmic reticulum together form cellular membrane system. Why the connections are there between the membranes of endoplasmic reticulum and the membranes of Golgi apparatus? We have learnt that the rough endoplasmic reticulum is the site of protein synthesis and smooth endoplasmic reticulum is the site of lipid synthesis. The material that is synthesized near the endoplasmic reticulum, it is packaged and dispatched to various targets inside and outside the cell through Golgi apparatus. So whatever the material that is synthesized at an endoplasmic reticulum, it has to be packaged and dispatched to various target sites. The target sites, some of them may be present inside the cell, some of them may be present outside the cell. Whatever it may be. For example, here it is a target site. Target 1, target 2, target 3, like that sites are there. Okay. And some of them are inside the cell and some of them are outside the cell. Material is synthesized at near endoplasmic reticulum. It is getting dispatched to target 1, target 2 and target 3. Some of them are inside the cell, some of them are outside the cell. Through what it is getting packaged and dispatched? Through Golgi apparatus. Through Golgi apparatus. Right. So, Golgi apparatus is having the functions of storage, modification and packaging of the products in its vesicles. So, as the material is entering in Golgi apparatus, before moving out, first it is getting storaged. Okay, storage. Next, after that it is getting modified. Next, it will get packaged. Then it will be moving out, dispatched. So these are the functions of a Golgi apparatus. So storage, modification, packaging and dispatching of materials is the function of the vesicles in Golgi apparatus. Sometimes some complex sugars are formed from simple sugars. Complex proteins are formed from simple proteins. Complex lipids are formed from simple lipids. Right? Is it the only function of Golgi apparatus or any other function is also there? Means, is modification the only function of Golgi apparatus or any other functions also? Golgi apparatus is involved in the formation of lysosomes. Golgi apparatus is involved in the formation of lysosomes. So from which organelle lysosomes are formed? Lysosomes are formed from Golgi apparatus. Right? Like how cells are formed from pre-existing cells, lysosomes being an organelle, it is formed from already existing organelle. So, lysosomes are called cells within the cells because cells arise from pre-existing cells. The same way, organelle, if it is arising from pre-existing another organelle, then it can be considered as cell within the cell. So likewise, which organelle is formed from the already existing organelle? Lysosomes. Lysosomes are called cells within the cells. Right? Coming to lysosomes. Lysis means breakdown. Soma means body. Lysis, breakdown. Soma means body. So lysosome, it is a body or the region which results in the breakdown of many molecules. Okay, so lysosomes are waste disposal system of the cell. Waste disposal system of the cell. So if you take unwanted plastic or unwanted any other material, if you want to throw it out, dispose it off, what will you do? Will you throw it out in its whole bulk size, or you will be making it into pieces? You will be making it into pieces. So if cell is having any waste biomolecules, unused biomolecules or worn out organelles are there in it, they are taken into lysosomes and are broken down into pieces or into their simpler units. Then they are thrown out. That's why lysosomes are known as waste disposal system of the cell. So the cell will be kept clean. How this particular activity is carried out? Lysosomes are involved in the digestion of a foreign material as well as worn out cellular organelles. They are involved in the digestion means breakdown. Digestion or breakdown of foreign material and what? Worn out cellular organelles. Worn out cellular organelles. Old. Right? So, the foreign material which enters into the cell and worn out cellular organelles, finally where do they enter? They enter in lysosomes in order to get disposed of. Before getting disposed of, what will happen to them? The foreign material and worn out cellular organelles will be broken down into small pieces. So lysosomes, how will they achieve this breakdown process? Lysosomes contain many powerful digestive enzymes which are capable of breaking down most of the organic material into their small pieces. If in any case major cellular disturbance occurs in the cellular metabolism and the cell gets completely damaged, lysosomes, they will burst out and release their hydrolytic enzymes. When these enzymes are released out, they digest their entire cell and cause the death of the cell. Thus, the lysosomes are also known as suicidal bags of the cell. So, lysosomes in the true sense, how can we consider them? These are the membrane-bound sacs which are filled with several digestive enzymes. And the enzymes present in lysosomes are made by rough endoplasmic reticulum. Right. So, the foreign material and worn out cellular organelles, they will finally end up in the lysosomes, and lysosomes, they break them up into small pieces by using the hydrolytic enzymes present in them. In a major cellular damage or cellular disturbance, the cell may entirely gets damaged. During that time, lysosomes will open out and release their hydrolytic enzymes in the cell, and these released enzymes cause the digestion of the entire cellular contents and the death of the cell. Thus, lysosomes are called suicidal bags of the cell. Lysosomes are called suicidal bags of the cell because in major disturbance of the cell, if the cell gets damaged severely, then lysosomes burst out and release their hydrolytic enzymes and cause the digestion of entire cellular contents entire cellular contents and causing the death of the cell. Causing the death of the cell. Causing the death of the cell. So only lysosomes are called suicidal bags of the cell. And structurally, what lysosomes can be considered as? Membrane-bound sacs that are filled with digestive enzymes. The digestive enzymes of lysosomes are made by which organelle? Rough endoplasmic reticulum. The digestive enzymes of lysosomes are made by digestive enzymes of lysosomes are made by rough endoplasmic reticulum. Right. Next, coming to the another organelle, mitochondria and plastids. Let us see mitochondria and plastids. Mitochondria these are known as powerhouse of the cell. Mitochondria are known as powerhouse of the cell. Right? Mitochondria. Actually, mitochondrion, it is singular. On, on. Singular. Mitochondria. It is plural. One is mitochondria. More than one mitochondria. These are what known as powerhouses of the cell. Powerhouses of the cell. Inside the cell, various chemical activities are carried out. All of them need energy. And this energy that is needed by the chemical activities of the cell, or the energy that is needed by the chemical activities of the cell, is produced from mitochondria in the form of ATP. The form of ATP. So the ATP is the form of energy. The full form of ATP is adenosine triphosphate. Adenosine triphosphate. Right. And as it is fueling all the chemical activities of the cell, like how currency is fueling all of our regular needs in our day-to-day life. ATP is known as energy currency of the cell. ATP is known as energy currency of a cell. Energy currency of the cell. New chemical compounds that are needed by the body in its construction using the energy stored in ATP. And mechanical work is carried out using the energy stored in ATP. Okay, so what for the ATP is used? It is used in common, what we can say, for chemical activities of the cell. the synthesis of new chemical compounds uses the energy stored in the ATP. Mechanical work uses the energy stored in ATP. If you take the structure of the mitochondria, mitochondria have two membrane coverings, outer covering and inner covering. Outer membrane and inner membrane. Two membrane coverings. The outer membrane is very porous. This one, it is very porous. Porous means it is having many pores in its structure. And inner membrane is deeply folded, like this, deeply folded. And whenever something is folded, it is going to achieve more surface area. More surface area. So the foldings of the inner membrane, they provide or they create a large surface area for ATP generating chemical reactions. So mitochondria produce ATP. Energy is produced by mitochondria in the form of ATP. That energy producing ATP generating chemical reactions going to have larger surface area by the deep foldings of the inner membrane of mitochondria. So there is a common point for mitochondria and plastids. After discussing plastids, I will let you know what is that common point. Both mitochondria and plastids are known as strange organelles. Why are they called strange organelles? Let us see first by discussing the plastids. Plastids are the special organelles which are present only in plant cells. Plastids, they are present only in plant cells. There are two types of plastids. Two types of plastids. One is leucoplasts. Another one is chromoplasts. Leuco means colorless. So these plastids, they do not have any pigment, lack pigments. So they appear colorless. Chromo means colored. Chromo means colored. So they have pigments or colored substance. Right? So these pigments will vary in different colors. One of such pigments is chlorophyll. The chromoplasts that contain chlorophyll are known as chloroplasts. What is chloroplast? The chromoplast which contain chlorophyll is known as chloroplast. The chloroplasts, they do contain some yellow or orange pigments along with chlorophyll. Okay, what is their significance? In 11th class we will learn. Do chloroplasts contain only green color pigments? No. Chloroplasts contain yellow and orange color pigments also. They do have their significance. What is the significance? We will learn in class 11th. Leucoplasts. So we know what for the chloroplasts are used for photosynthesis. And what for the leucoplasts are used? Leucoplasts, they store starch, oils and protein, etc. Leucoplasts, they store the starch, protein and oils are lipids, right? Starch storing leucoplasts are called amyloplasts. Protein storing leucoplasts are called aleuroplasts, and oil storing are called elaioplasts. Coming to strange organelles. Okay. Coming to the structure of plastids. Like how mitochondria have two membranes, plastids also have two coverings. Inside the plastid, there are many membrane layers embedded in some colorless material. That colorless material is known as stroma. And plastids and mitochondria are called strange organelles because they have their own DNA and some ribosomes. No other organelles have like this. That's why plastids and mitochondria are called strange organelles. Okay, thank you.